Good day to the chairman of the conference, delegates, adjudicators, and viewers. My name is Kayla Pillay, and I will be taking you through my PhD project titled Phenotypic and Genotypic Diversity of Gardnerella vaginalis isolates from South African pregnant women. To begin, I'd like to take you through the scheme of the study, which involves the background, aim, objectives of which there are 3, methodology and results for each, the discussion, limitations, and conclusion. To provide a bit of background on Gardnerella vaginalis, G. vaginalis is a gram-variable pleomorphic coccobacillus and is the predominant pathogen responsible for the onset and progression of bacterial vaginosis, which is a polymicrobial syndrome resulting from the replacement of the native lactobacilli in the vagina with anaerobic commensals. The commensal and pathogenic nature of G. vaginalis warrants the need for understanding pathogenic mechanisms that this microorganism uses to convey its virulence. The diversity of G. vaginalis has not yet been explored in a South African setting. If untreated, bacterial vaginosis in pregnant women causes pelvic inflammatory disease, preterm birth, increased susceptibility to STIs, and may result in maternal or neonatal morbidity and mortality. To provide a bit of statistics, the global prevalence of BV is 23 to 29% among women of reproductive age. In pregnant women, it is reported to be between 8 and 51%. The aim of the study is thus to investigate the phenotypic and genotypic diversity of G. vaginalis isolates in South African pregnant women diagnosed as BV positive, BV intermediate, and BV negative. The first objective of the study was to determine the prevalence and risk factors associated with BV. Ethical approval was obtained for the study from the Biomedical Research Ethics Committee of the University of KwaZulu-Natal. 150 pregnant women were enrolled in the study and were tasked to provide 2 self-collected vaginal swabs. The first swab was used for BV diagnosis, and the second swab was used for G. vaginalis culture and further lab testing. Risk factors were determined by statistical analysis of structured questionnaires administered to the study participants. Prevalence was measured based on BV diagnosis by Nugent scoring from gram-stained vaginal smears. Table 1 represents the baseline characteristics of the study population stratified by BV status. From the table, we see that the overall population was unmarried, completed high school as the highest level of education, did not report symptoms of abnormal vaginal discharge, had regular sex partners, did not live with their sex partners, had sexual debuts between the ages of 15 to 20, had 2 to 4 lifetime partners, did not use a condom during their last sexual act, reported condom use only sometimes, had uncircumcised partners, and did not know if their partners had other partners. They were also in the 3rd trimester of pregnancy. Table 2 is representative of the BV prevalence in the study. 22% of BV prevalence, was 22% in BV positive women, 28% in BV intermediate women, and 49.3% in BV negative women. The second objective of our study was characterization based on biochemical and genetic composition. This was achieved through phenotypic and genotypic characterization of the G. vaginalis isolates. Phenotypic characterization was conducted through biochemical tests that included beta hemolysis, lipase, and hipurate hydrolysis. Catalase and oxidase was also performed for the identification of G-vag. Biotype assignment was according to Pio et al. 1984. Table indicates the methods used for each biochemical test. Beta hemolysis was done using tubes containing 0.1 ml of NaCl inoculated with a 48-hour fresh culture of G. vaginalis. ONPG discs were added to the tubes, and the tubes were incubated at 35 degrees for 4 to 6 hours. A positive result was indicated by a color change in the medium to yellow. For lipase, a single colony streak was made on egg yolk medium egg plates. Following sterilization, 10 moles of egg yolk emulsion and 5 moles of horse serum was added to the medium. The plates were incubated at 36 degrees for 3 days in an anaerobic jar. Once incubation was complete, the plates were observed for an oily iridescent sheen around colonies, which was indicative of a positive test result. Purate hydrolysis was done using 1 to 3 colonies of fresh isolates emulsified in small plastic tubes containing 0.1 moles of sterile demineralized water. Purate discs were added to each tube, and the tubes were incubated aerobically for 2 hours at 35 to 37 degrees. Following incubation, 2 drops of non-hydrogen reagent was added to the suspension and re-incubated for 30 minutes at 36 degrees. The blue to purple color change indicated a positive result. Genotyping was conducted through amplified ribosomal DNA restriction analysis, or the ARDRA method. Genotypic assignment was based on restriction fragments obtained upon digestion of the 16S rRNA amplicons with TAC1 at 65 degrees for 3 hours. Products were analyzed on a 1.5% agarose gel with TBE buffer. From the 150 vaginal swabs, 17 isolates of G. vaginalis were successfully isolated. This may have been due to a loss of isolates in the culture process due to improper culture techniques, introduction of contamination, or incubation periods. Table 3 indicates the phenotypic characterization of the 17 isolates based on the results obtained from each biochemical test. The corresponding biotype of each isolate is indicated in the table. The table also shows that 5 different biotypes were identified: biotypes 1, 2, 3, 5, and 7, with biotype 3 being the most prevalent, accounting for almost 50% of isolates, followed by biotype 5 and biotype 7. 2 isolates were classified as biotype 2 and 1 as biotype 1. Biotypes 4, 6, and 8 were not found in our study. Only 16 isolates were further used for analysis, as GV3 was disregarded as G. vaginalis following unsuccessful amplification and sequencing. Figure 1 represents the genotyping profiles that were achieved using TAC1. 4 genotypes are indicated, as can be seen in the diagram, with corresponding band sizes represented in Table 4. The genotypes associated with each isolate is also represented in Table 4. Genotyping analysis revealed the presence of 4 different genotypes circulating within our population. Genotype 1 right up till genotype 4, as indicated in Figure 1. Genotype 1 was the most prevalent genotype in 56% isolates, or 9 out of 16, followed by genotype 3, genotype 4, and genotype 2. In the BV positive and intermediate women, genotype 1 was dominant with 60 and 56% respectively. Genotype 3 had a prevalence of 40 and 22% respectively across the BV positive and intermediate states. The BV intermediate state also included genotype 2 and genotype 4. The BV negative woman harbored genotype 1 and genotype 4 in equal amounts, as can be seen in Table 4. The final objective of our study was the association of genotypes and biotypes with symptoms of BV and BV status. Phylogenetic analysis was conducted based on the phylogenetic tree, which was compiled using sequences. The phylogenetic tree enabled us to create or identify associations based on baby status, genotypes, and biotypes. The associations are depicted in Table 5. The phylogenetic analysis revealed heterogeneity in the gene clusters. This means that isolates from BV positive and intermediate states and isolates from BV positive and negative states were shown to cluster together. The most common genotype-biotype combination was distributed across the BV intermediate and negative states, that is, genotype 1 and biotype 3, accounting for 1/4 of the isolates. The different gene clusters observed indicated that a low level of diversity existed within G. vaginalis isolates from across the different BV states. Within genotype 1, biotype 3 was dominant and majority of the women were BV intermediate. They had also reported symptoms of abnormal vaginal discharge. The only woman harboring genotype 2 was classified as biotype 3 and reported no symptoms of discharge. The composite genotype 3, made up of band patterns corresponding with genotype 1 and 2, had biotypes 3 and 5, were from BV positive and intermediate groups, and had equal amounts of women reporting symptoms of discharge and no symptoms of discharge. The women harboring genotype 4 were BV intermediate and BV negative in equal amounts with biotypes 3 and 7. Both women had reported symptoms of abnormal vaginal discharge. To take you through our discussion, 22% BV prevalence was reported. This is consistent with the findings of BV prevalence in other studies conducted in pregnant women across the African continent, in northeastern Nigeria, South Africa and Kenya, and Ethiopia, and is comparable with the 24.8% combined prevalence report of BV in Africa. A significant association was identified between the presence of abnormal discharge and BV status. Among 16 isolates, most of the BV-positive pregnant women presented with abnormal vaginal discharge. This result supports a similar study conducted by Ahmed et al., in which pregnant women who reported vaginal discharge were significantly associated with BV. The most prevalent biotypes were biotypes 3, 5, and 7, accounting for 82% of the total isolates. The most prevalent biotype, biotype 3, was found in the BV negative and intermediate women. The findings of this study are consistent with the findings of a study conducted by Nisha et al., which stated that biotype 3 was more associated with non-BV than BV. Based on the biotype distribution observed in our study, a difference can be observed in isolates obtained from women with and without BV. The current study also reports 2 additional genotypes, genotype 3 with band sizes 250, 360, 400, and 500 base pairs, and genotype 4 with band sizes 250, 400, 500, and 800 base pairs. This is a significant finding and significant progress because the findings by Plecatide et al. Had reported only 2 genotypes, and Dos Santos Santiago et al. Had reported 3. Symptoms associated with BV, that is, abnormal vaginal discharge, was observed across women diagnosed as BV positive and intermediate. Most of the asymptomatic women harbored genotype 1 and 3, and a higher proportion of these women were diagnosed as BV positive and intermediate. This result is inconsistent with the Dos Santos Santiago study, which showed genotype 2 to be the most prevalent genotype associated with symptomatic BV. Our study was also limited in some ways. The small sample size of cultured isolates did not display the diversification in its entirety, and we may have missed significant association amongst isolates across the BV groups. A control group of non-pregnant women was not introduced for comparison studies of genotypes and biotypes. The cross-sectional nature of the study restricted us from developing associations of genotypes and biotypes with the acquisition of other STIs. In conclusion, biochemical testing revealed that biotype 3 was the most dominant within the study population. Genotypic analysis revealed the presence of 4 genotypes circulating within our population, of which genotype 1 was most dominant and significantly associated with symptomatic BV. Future studies should be focused on understanding the pathogenesis of G. vaginalis and its impact on female reproductive health across all populations. Although a low level of diversity existed between isolates and across the BV status, across all BV states, more work is needed to establish the exact mechanisms through which BV conducts its pathogenesis. This study provides the first report on the diversity of G. vaginalis isolates across the different BV states in South African pregnant women, while establishing a link between genotypes and biotypes with symptoms of the disease. The following references were used in the study. I would like to acknowledge the National Research Foundation, the University of KwaZulu-Natal, and the Clinical Medicine Laboratory at the University of KwaZulu-Natal. Thank you.